So why did the dinosaurs go extinct, but then other archosaurs like crocodiles and birds didn't?
So that's in a sense, a very challenging question
Yeah.
because, the answer would be there's a sort of simplistic answer and a much more complicated answer. And the short answer is there were a lot of ecological changes in the late Cretaceous era
Yeah.
when, dinosaurs ruled the earth, as we learned from Jurassic Park or Cretaceous Park, I suppose it should be. And at the end of this, sort of the coup de grace was probably the major impact event that everybody heard of. Big meteor hits the earth, enormous ecological changes, and the majority of dinosaurs went extinct, but a few lineages of dinosaurs, those we call birds, made it through.
Mm.
What exactly um, permitted this is still a little obscure. I mean, in general, smaller organisms survive mass extinctions much better.
Yeah.
This is something called the Lilliput effect. So some of it may simply be the size distribution, but there were a lot of small non-avian dinosaurs as well. So
Mm.
there may have been a component of luck.
Mm.
Um, but people say that, the end of the Cretaceous was the end of the era of dinosaurs, but I like to point out that there are many more species of dinosaur than there are of many more species of birds, dinosaurs, than
Yeah.
there are mammals. So,
Yeah.
you know, it's still the age of dinosaurs.
Yeah. 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 And what about the crocodiles? Is it
So,
crocodilia?
and that's
Dilians.
also another hard case because, um, obviously they were very ecologically different. They weren't
Yeah.
small. And why did they make it through? Now, one thing to recognize about crocodilians is they're not very biodiverse, and they
Mm.
were more biodiverse in the past.
Yeah.
There's about 24 named species right now of crocodilian. There's actually a wonderful zoo in Florida where they have exemplars of all species. So
Nice.
if anybody's coming to St. Augustine, go see that.
Yeah. And there's some theories that I've read about where for the birds it was potentially because well, for the beaked birds, because apparently there was tooth birds with teeth. Am I
Yes.
correct in saying that? I did not know that. That's really crazy. But didn't they feed the beaked birds fed on seeds, which would have been probably available after
Yeah.
the asteroid hit? Is that one of the theories, right?
It's certainly a theory, and I tend to be fairly cautious about attributing uh, exact reasons to the survival of, you know, taxa with different phenotypes. And
Mm.
there almost certainly was some stochasticity in the sense of just luck.
Yeah.
Um, and one of the interesting things I will say that we found from genetic evidence is that there was probably at least some lineages of extant bird where there was a great increase in the population size. Presumably, what was happening is a lot of ecological space was opened up
Yeah.
because of the mass extinction. And I was actually a little amazed that we could see the signal of this. And I can go a little later into the details of how we see the signal, but
Mm.
there's still a genomic signal, a genetic signal in the bird's genomes for certain lineages that they increased in their population size for a period. And then,
Mm.
as evolution occurred and ecological niches filled up with new taxa, things calmed down a little bit. So,
Mm.
so we see still in bird genomes the signature of the mass extinction at the end of the Cretaceous Paleogene. boundary, which is essentially the point where the non-avian dinosaurs went extinct, and the dinosaurs we still have continued living on.
Mm. Yeah. And then I read something about another like theory about the crocodilians. It was because they didn't rely on food to regulate their body temperature, so they could go a longer period without eating anything. Could that be a potential reason as well as luck?
Yeah, and certainly, luck, fortune favors the prepared, I
Yeah.
suppose.
Yeah.
So, if certain physiologies were probably predisposed to making it through the Cretaceous Paleogene mass extinction, um, but an interesting thing is, of course, that you can probably look back and say, oh, there were other taxa that had, similar physiologies. Um, why didn't they make it? And of
Mm.
course, we also have to recognize that a major component of the luck is where did the asteroid hit?
Yeah.
Certainly, I don't want to make it sound like the current hypothesis is that the only thing was the asteroid. Um, there were ecological changes in the Upper Cretaceous, and certain there was turnover of a number of lineages. So the world was changing. The world is always changing,
Yeah.
and you know, we had sort of a coup de grace, but no doubt, um, taxa that were distributed in certain parts of the world fared much better. You know,
Mm.
having the, you know, asteroid impact right next door was not a good day for anybody.
Yeah. Yeah. And what areas of the world from the
so the
research
chick,
fared best?
so the Chicxulub crater was in sort of Central America, or what would be Central America right now. Now, of course, what happened with an impact of that size is a lot of ejecta gets, uh, blasted off into space, and a fair
Mm.
amount of that comes down. So probably the antipodes of, the opposite side of the planet from that also suffered a bit as things um, started to rain back down. Um, the short answer, of course, is that reconstructing biogeography that deep is challenging. So,
Yeah.
um, a number of years ago, I was part of a group that published a paper trying to reconstruct as far back as we could go the biogeographic movements of uh, what I like to call the barnyard birds, the chickens and their relatives. And, fundamentally, as you get deeper and deeper in time, unsurprisingly, uh, it becomes harder and harder to reconstruct. And of
Mm.
course, the continents are moving. Essentially, we're trying to work with a moving target using, the best computational tools we can use to figure this out. And I think a fair assessment becomes, well, we're uncertain. Um, but clearly, the barnyard birds were a group of birds, and I sort of use that obviously a little jokingly, but this is a clade. A clade is a defined group of organisms with a common ancestor.
Yeah.
This is a clade that comprises the so-called galliform birds, which are chicken, Gallus gallus, and many of its relatives, quail, turkeys, grouse,
Mm.
um, many others, and the ducks and geese. the anseriform birds. So this is a large group of birds. And actually the Galloancerae is the which is the clade comprising both of those groups, are one of the lineages where uppermost Cretaceous fossils have been found. And I'll give a shout out to Daniel Field, the paleontologist, uh, one of the paleontologists who worked on that, as well as Dan Sepka, the Dans. And I've been lucky enough to work with both of them to bring that paleontological perspective.
So where did the chickens originate from what you found?
So the Upper Cretaceous fossil was from Europe. Um, so they probably had a fairly cosmopolitan uh, distribution.
Yeah.
One hard thing about avian biogeography, especially going far into the past, is of course birds fly.
Yeah.
So they're a lot more mobile. And even if you look at the flightless birds of today, things like the ostrich, the emu, the kiwis, and so forth, and not every flightless bird, but this major group called the Paleognace,
Mm.
which include the ostrich, emu, cassowary, rheas, and so forth. A hypothesis for a long time was that that group was very ancient. And there's almost a poetic vision of how they got where they are in the earlier theory, which is as the continents broke up, as the supercontinent of Gondwana, the southern supercontinent, as it broke up, these large flightless birds sort of rafted apart, which I think was a poetic hypothesis. Unfortunately, um, Thomas Huxley had a famous saying that many a beautiful hypothesis is slain by some ugly data.
Yeah.
And what we found when we looked at the genomes of these birds was a very clear signal that um, there is a volant group, a group of birds of paleognath birds that could fly, the tinamous, that are nested within the flightless lineages.
Mm.
So what that really says is, um, well, there are two ways to explain this, one much more plausible than the other. The less plausible hypothesis was they lost flight and that the tinamous regained flight. That seems gaining flight is hard. So the hypothesis that is almost certainly correct is that many different lineages of Paleognace lost flight independently. So they were
Mm.
probably much more mobile um, in the sort of early Paleogene. So immediately after the extinction of the dinosaurs, and no doubt a little before, they were much more mobile. They were volant, and so they could fly, they could move around. Than well, than they are now, where they can't fly. And
Mm.
indeed, when people have, and I will give full credit, this was not my work, but related work building on some of our knowledge of Paleognath phylogeny. When people looked at the genomes of fossil elephant birds, which are on Madagascar, they turn out to be most closely related to kiwis.
Wow.
So these birds were flying around
Yeah.
um, and quite mobile. So reconstructing their biogeography is you know after finding all the data we found, I felt like, well, in some ways it's almost a fool's errand.
Mm.
They flew around a lot, And when they found environments where they lost flight, I'm anthropomorphizing, I suppose, a little bit. When the lineage, when ancestors of these groups ended up in places where the loss of flight was possible,
Mm.
um, they lost flight. And those are the ones we see now. So far from this poetic vision of, these giant ostrich-like things rafting apart on the continents as they slowly drift apart. It's actually these guys could fly, and they distributed themselves. And what we see are the relics of this lineage that distributed itself around the world. So,
Mm. And is the common ancestor of all of these the one that could fly? Do we
oh,
know how big? Do we know how big it was compared to an ostrich or chicken today?
so it was certainly much smaller.
Yeah.
And I will here again give a shout out to Daniel Field and some of his colleagues who had a very recent paper on the topic, which I'm not going to summarize because it was so recent it crossed my desk and I've only read the abstract. So apologies for that one. And that really sort of refined it. But it was pretty clear, I would say, that the taxa that were volant and could move around, the common ancestor of these, is much smaller.
Mm.
Um, Certainly, even if you made stronger wings on an ostrich, there are sort of limits.
Yeah. Yeah.
So,
Definitely.
that was a a different time in Earth history. And one of the things I will say that always fascinates me about biology. So when I think of physics as a science, and I look to physics some because one of my major interests is reconstructing the history of genomes with computational models. Um, so I look to that, but physics is in some ways a universal science. Many of the laws that we see will apply just as well here
Yeah.
as in a galaxy far, far away. Um, Whereas biology is historically constrained. The fundamental hypothesis of biology is a branching process where new species are uh, where lineage is split, form new species. Sometimes there is gene flow. If we think of humans, of course, uh, Europeans and Eurasians more broadly tend to have some Neanderthal ancestry. So there was a split, and then, of course, we met again, and gene flow occurs. That happens across the tree of life. Certainly in birds, there are many examples, but by and large, the unifying theory of biology is essentially we have ancestors, they split, um, become genetically isolated from each other, speciation process.
Yeah.
And because of that, there is a important historical contingency component to evolution. And what I mean by that is um, we're constrained by the phenotypes and genotypes of our ancestors. So, what our ancestral genomes are determine to some degree what's possible.
Mhm.
Um,
Yeah.
so, um, Universal laws are hard to find in biology. Mhm.
Mm. Yeah. Especially because there's so many different species and lineages as well. So it could be really dependent. The history can be different for each one, right? So that makes it way more confusing.
Oh, yes, yes. Obviously, um, the simple fact that the genetic machinery um, is changing over time makes what's possible different in different lineages.
Mm.
I mean, in some ways, it is still rather amazing when we trace genomic histories, the things that are possible in terms of changes. But, um, for example, we're not going to suddenly, uh, our descendants, assuming, humans leave descendants 20 million years from now, are unlikely to suddenly become photosynthetic. I suppose unless we do some genetic engineering, but even
Yeah.
that's a little, that moves much faster, artificial life. But
Mm.
just in terms of normal trajectories, the genetic machinery for certain things simply doesn't exist in certain lineages.
Mm.
Um, so that's an important component to realize. Mutation doesn't work like, the X-Men, where all of a sudden a really interesting power suddenly appears.
Yeah. Yeah, because as far as I'm aware from A-level biology, genetic mutation is random, right?
Yeah.
So but how drastic can the mutation be? And isn't most of the time the mutations not actually necessarily adaptive or advantageous? A lot of the time it can be the opposite, right?
Oh, yeah. So there's really sort of two major types of mutations, I would say. Um, And actually, if you think of much of our genomes and I'm now using humans and other vertebrates, much of our genomes are not functional.
Mm.
Um, the word that's often applied is junk DNA. That's gotten some criticism, but I think at the end of the day, it's ultimately correct. In fact, mutation tells us it's correct in the sense that the mutational burden, if you imagined all of our genome being functional, the mutational burden would just be too much.
Mm.
Now, mutations in a lot of these areas are so-called neutral mutations, where they do not affect the fitness of the
Yeah.
organism. Neutral mutations are um, their their history is dominated by genetic drift, by random chance, and
Mm.
that's a major rule. For things that uh, for other mutations, um, it's much more likely to have a deleterious mutation than to have an advantageous mutation.
Mm.
Now, with that said, um, what's advantageous and what's deleterious is also environment dependent.
Yeah. Yeah.
So, um, in one environment, as well as to some degree, what's neutral versus what's deleterious is environment dependent. I mean, humans need vitamin C.
Mm.
Many mammals do not. That's because they have a gene that can synthesize vitamin C. If you look in the human genome, you can see a segment of DNA that looks like that gene,
Oh,
but it
really?
has
Wow.
inactivating mutations.
Oh.
So our ancestors probably got enough vitamin C in their diets. And if you suffer a mutation in a gene that makes something that you can get in your diet, genetic drift can fix it. And it
Mm.
wasn't until, humans started getting on long range, boat trips when they
Yeah.
found they had to bring along, citrus to to avoid the scurvy, that the vitamin C, the inability to synthesize vitamin C became problematic.
Mm.
Um, and, you know, in birds, for example, the uh, sweet taste receptors, for example, have evolved multiple times from other taste receptors, um, rather than, sort of having been preserved. So, um, what what is probably selecting for that? Well, some of these lineages, for example, have become frugivorous or something. They eat fruit, or, you know, things like that. So as mutations happen, they may alter the whole landscape of what's possible for phenotypes.
Mm,
Um, so a simple mutation might open up a huge number of possibilities.
mm. And what are some pivotal mutations in the human lineage that we're aware of, if
Ah,
any?
and now you're reaching into humans where, so, obviously one major mutation. I mean, if we think about what makes humans to some degree human, it's of course up here, our
Mm,
brains. Um, Certainly the primate lineage overall is a big brained lineage, but there were certainly mutations in in our evolutionary history that allowed our brain to expand in size. Um, and certainly cognitive abilities and so forth are not absolutely tied to the exact size of your brain. And variation within humans is you know modest. We're all human. But the expanded brain sizes certainly mean something. Certainly, they expanded our cognitive abilities, and indeed, we see certain lineages of birds having similar brain expansions. A number of years ago, we did a large scale, and this was led by one of the paleontologists I mentioned earlier, among others, Dan Sepka. Um, who um and I want to make sure to give credit where credit is due. I'll just say science is a team sport. If I miss anybody, I will apologize in advance.
no
But
worries.
all of your listeners should know that science is a team sport, that we
Yeah.
all work together, we trade ideas, if we didn't all work together and have colleagues who had really good ideas, science wouldn't move. But
Mm,
back to the big picture of the actual science, with apologies to everybody who contributed to that effort, we can certainly see certain lineages where there have been shifts in relative brain size. And
mm,
you can probably guess the birds that have shown expanded brain sizes over evolutionary times: the parrots,
mm. Yeah.
corvids, so
Yeah.
crows, incredibly smart, incredible
Yeah.
problem solvers. Um, so, uh, is the genetic architecture underlying that um, similar to humans or fundamentally different? The answer is we don't completely know. Certainly, we're going to find some differences. It's very uh, hard for the tape of evolution. If you videotaped our evolutionary history going back many years, Stephen Jay Gould had a famous. If you replayed the tape of history, would it be the same? And the short answer is no.
Mm,
Stochasticity. Who
oh,
survives a mass extinction? Um, what specific mutations happened? Uh, I talked about neutral mutations and dominating uh, what we see, and that's true. But our phenotypes ultimately are related to the other mutations, the ones that altered fitness. They're ultimately random.
mm,
Um, Now, obviously, there's a selection on those. We do not see the deleterious mutations. If their natural selection weeds them out.
Yeah.
Um, there's a little complexity there that I won't go into because, how strong natural selection is depends on the population size. But
Yeah.
um, uh, which actually, maybe I should go into that just for a moment because this gets at an important idea in evolutionary history, which is uh, the strength of natural selection depends on the population size. In small populations, deleterious mutations may not be, the population size may not be sufficient for selection to overwhelm drift, which of course creates this problem. We live right now in a mass extinction. Unfortunately, a mass extinction that we're causing by expanding across the world and so dominating resource utilization. We're the asteroid that's hitting the Earth. Um, but it's not just preserving some organisms. We have to preserve viable populations. And um, because, if we had a population size that any group of organisms had a very small population size, they could go into sort of a death spiral where new deleterious mutations, um, uh, selection does not overwhelm drift. But of course, new deleterious mutations cause lower, I was about to call it hatching success. You can tell that I think about birds a lot.
Yeah. Yeah.
um, you have fewer new individuals entering the population, which causes the impact of genetic drift to be even worse, and death spiral, organisms go away. So I think that's something that, hopefully everybody should be thinking about, that, we can't just preserve two of each animal
It's not Noah's
in
Ark.
a zoo. We can't do Noah's Ark.
Yeah.
There has to be sufficient genetic variation and
Yep.
sufficient population sizes for a group to be viable. So,
Mm.
you know, uh, I feel like now I've moved into some depressing territory, talking about humans being the asteroid hitting the Earth. So,
Yeah. No, it's all good. And why have birds evolved so much faster than the crocodilians? You did a paper or project on the crocodilians, and you said that you and your team said that they've evolved quite slowly. Why is there a difference between these two?
so that's actually a really good question, and certainly um, a big component is actually generation time.
Mm.
So mutations that show up in future generations are so-called germline mutations. They're happening in the cells that create sperm or eggs, and um, ultimately they happen on a per generation basis. Now, with that said, over superimposed on that are sort of some metabolic features of of sort of the life history, and indeed, you know birds on a certainly on a per million year basis have evolved a lot faster than crocodilians. Crocodilians and turtles are among the lowest mutation rates over in terms of accumulated substitutions in the genomes over time. But a lot of that is probably um, generation time.
Mm.
Now, For example, if we were to go into mammals, the muroid rodents, things like mice, have an incredible mutation rate. Now, obviously, have a very short generation time, but in terms of comparable generation time, they actually are experiencing more substitutions over time. Um, and I'm sort of flipping back and forth between mutation and substitution,
Mm.
and I should make sure to define the difference.
Yeah, yeah.
When we talk about a mutation, ultimately that's something that's happening in an individual, and if it's happening in the germline, I mean, if I have a mutation in a cell in my arm, it's just a mutation in a cell in my arm. It's never going to be passed along.
Yeah.
But if there's a germline mutation and it's passed along, that's still a mutation that occurred in an individual that has now entered the population.
Mm.
When we talk about substitutions, and a lot of what I study, because I compare genomes for different organisms, um, are substitutions, which are mutations that have entered the population. They have not been culled by natural selection. They have not been lost by due to genetic drift. And obviously, it's a uh, every new mutation that enters the population that's only entering, a few individuals. It may it may increase in frequency and eventually take over the population. Then it would be a substitution, using the terminology I'm giving out. But mutations are very broad. And indeed,
Mm.
one of the really cool things modern genomic uh, methodologies have done for us is the ability to directly estimate mutation rates. Um, Essentially, you take mom and dad, you sequence them, take one of their kids, and you sequence the kid, and you say, okay, I can match up the genes here to the father's genes or the mother's genes, and whoops, here's a base pair that I can't find in either.
Mm.
There's a lot of, devil in the details because, of course, sequencing is not absolutely accurate. And probably the ideal way to do that is, um, sort of mom, dad, one of their kids, that kid's mate, and one of the grandchildren,
Yeah.
um, and sort of do a three generation. And in fact, I have some three generation data or I and collaborators have a three generation data set for particular lineage that unfortunately is not yet analyzed.
Mm.
But, we're starting to get some real clues as to both the rate of new mutations and the spectrum of new mutations. In other words, what kind of base pair changes are more likely and less likely?
Mm.
Um, and there is some theory that would say that at least for the subset that are neutral, because drift is dominating the um, uh, how they're passed on in populations, we should see something very similar to the input of new mutations if they're occurring in a region where um, they don't have a possibility of impacting function. Now, obviously, if in that fraction of the genome that is functional, if mutation happens that causes a loss of function that might be deleterious, or for that matter, a gain of function that might be deleterious, something that increases the activity, say, of a protein, but in a way that's not appropriate, um, then natural selection removes that and we don't see them. So
Mm.
um, I think that's the interesting thing. You started out by asking about rate in crocodilians versus birds. I actually believe, based on some of what we see, that a major difference is really just generation time. Takes a lot longer for the average um, crocodilian to become sexually mature
Yeah.
than the average bird. And indeed, what birds have a really low mutation rate? Things like albatrosses
Mm.
and so forth. What are the most long-lived birds?
Yeah.
So, and the ones with the longest generation times.
Yeah.
So it's not perfect, and there's no doubt some physiological changes altering mutation rates, but that's a biggie.
Yeah. And does this make sense? If humans, I'm going back to humans again, sorry. If humans are living longer, I know the age of sexual maturity remains relatively constant, but if less people are having children and that trend continues and people live longer, would that also slow the rate of our mutations compared to previous times in history where maybe people would have kids younger and have more kids and have them faster?
So there's an interesting aspect to this. So the short answer is it's probably not, if we could imagine, being the time traveler from H.G. Wells going into the future, and if that time traveler could also sequence the genomes of our descendants,
Yeah.
we probably wouldn't see a huge impact. And one reason is actually something that I'll just bring up, called male-driven molecular evolution. So, um, certainly, as females have children later in life, there are higher probabilities of certain problems occurring due to, for example, chromosomal abnormalities. But the number of cell divisions up to leading up to an egg is relatively constant.
Mm.
In contrast, you and I, our germ cells, our sperm, are being produced throughout our lives.
Yeah.
And since mutations are happening through cell divisions, older males will have more mutations in the sperm.
Mm.
So changing the sort of calendar for having children, at least in organisms, where over time, we have a very big separation between, birds or turtles, and birds and crocodilians, or birds and turtles, for example. Then, things that are going on due to sociological changes. Um, and of course, we don't know how long the sociological changes will
Yeah.
occur. I mean, sometimes uh, I reflect on, I think all the time about things that are occurring over time spans of millions of years.
Yeah.
And then I sit back and contemplate, the history of humans. And it's sort of like, wow, I'm going to a place in a relatively young country, the United States.
Mm.
Of course, there were natives here before people who looked like me came. Although my family came in the 20th century, so I take no blame for anything that happened
Yeah.
before then.
Yeah.
Uh, I might take some blame for things that happened in Europe, but who knows? Um, but joking aside, you know, I think back to locations in the US, and it's like, wow, these are ancient. And then I think, actually, they're not that old.
Yeah.
And compared to the timescales that I think about all the time when numbers come out of my computer, where I'm going, oh yeah, 50 million years.
Yeah, yeah, yeah.
So I think what's happening uh, ultimately is uh, over time scales, like, what's the changes that have happened sociologically in the 20th and now 21st centuries have been truly remarkable in one level, but um, they may just be a blip. In fact,
Yeah.
unless something happens that keeps certain trajectories going along, they're going to be a blip in evolutionary time. You won't even see it. I mean, future geologists will be able to see a layer of all the plastics we spread across the earth.
Yeah.
But I don't think future genomicists, if there are future genomicists, unless other things change, will be able to see an impact of 20th and 21st century sociological changes in our genomes. So,
It's humbling really to
yeah,
realize how slow we change genomically compared to as a society, and it puts things into perspective. It puts it you can have a positive spin on it as well, like you were saying about the situation with the world and things like that. It might cause a lot of harm, but even if we get wiped out, if there's something that remains,
yes.
expand the time horizon, like things will hopefully be restored again if we can't fix it ourselves. Hopefully this is just a blip and we can mitigate all the stuff that's happening.
If Star Trek has taught me nothing, it's that humans will come together and somehow solve these
Yeah.
problems. Of course, it's fiction, but
Yeah.
but I like to think that message is correct. Um, and the other thing I will say is, I joked earlier about us still being in the age of dinosaurs because there are more species of bird than there are of mammal.
Mm.
Um, but in reality, we are in and throughout all of Earth history, have been in the age of prokaryotes, of
Mm.
bacteria um, and archaea, the other major lineage of prokaryotic organisms without a nucleus. So, and I suppose, although I'm primarily a genomicist who focus on birds, I also have a fascination with bacteriophages that I've gone so far as to occasionally teach a small class in bacteriophage biology. And they're on the order of 10 bacteriophages. These are viruses that infect, bacteria. Um, that there are probably about, 10 bacteriophages for every bacterium in the world. So
Wow.
I guess in reality, we are always in the age of bacteriophages, and there is nothing humans can do to the planet that will wipe out the bacteria or the bacteriophages.
Yeah.
So um, that I suppose is also humbling to
Yeah.
to think when I've taught lab versions of this course as well, and one of the things I would have students do um, is go out in, a little wooded area and pick up, a tiny amount of soil. And um, then if you pass liquids through a zero point 0.2 micron filter, that's small enough that bacteria won't move through it. So you can sort of sterilize material by pushing it through a very small filter. Viruses, like bacteriophages, are small enough to go through.
Mm.
Um, and so I would have the students, soak it in something that would be, sort of nice for the bacteriophages to survive. push it through one of these filters, and then infect a strain of bacteria that we had in the lab. A very safe strain. No, no pathogens.
Yeah,
I'm not unleashing
the plague and the Black
students
Death.
on any pathogenic viruses or bacteria. But you could always, just in a tiny sample of soil, identify phages that would infect a strain of bacteria. So, it's incredibly humbling that, there are multitudes in the tiniest bit of soil. So,
Mm.
um, but uh, I will say that, like I said, I'm a computational biologist, an evolutionary biologist. I think about genomes. Uh, the genomes of phages are fascinating, but most of my actual work has been in the genomes, largely of vertebrates, a little of plants, some in yeast, um, but largely vertebrates and largely birds, and
Mm.
to some degree crocodilians.
Mm.
So um, I'm going to bring myself away from waxing poetic about the multitudes in a gram of soil.
Yeah. Well, there's a whole world in it though. There's a whole almost universe in something that
Yes.
we take for granted. We got a bit of dirt on my shoe, put that under a microscope, what are you going to find? Quite a lot of
Yeah,
biodiversity, right?
very much. And certainly everything from things you can see, like under very low magnification, to or relatively low, um, to you know uh, the bacteria, fungi, and then of course the phages and so forth. So the world biodiversity. I tend to think, like many people, about biodiversity in terms of large um, animals and plants that we can see.
Yeah.
Um, and I understand why people tend to think of it that way. It's a lot easier to sell an endangered panda.
Yeah.
We need to keep them around than some very tiny little creature that nobody can see. But um, there's incredible diversity that much of which we don't even completely know
Mm.
at the level of microbial life. Um, and I will say that I have a student who is trying to do some comparative work using genomic data from algae. And one of the things we always reflect on is, well, especially for some of these unicellular species and so forth, um, how well are they actually even being identified?
Mm.
Certainly there are experts, and I have collaborated with phycologists, people who study algae as well. Um, but um, it's just very hard for us to distinguish among these taxa. Unlike, for example, birds, where um, uh, especially people who are very good with birds. And I will freely admit that although I feel very good about bird genomes, um, I'm rather poor as an actual birder.
Really?
Some of that is one of my eyes is not very good, so I'm always doing binoculars like they're monoculars, sort of.
Yeah, yeah, yeah.
And especially as I've aged, my high frequency hearing has
Mm.
gotten worse, and I have been out with places where people have heard the song of birds that I know have a very high frequency song, and I'm like, can't hear a thing.
Wow.
So
Yeah.
now, most of the time I hear stuff.
Yeah.
It's not like I don't hear the owls around my neighborhood making a ruckus. I happen to be lucky enough to live across the street from an area that's kind of wooded, and it's an area you really couldn't build anything on because of drainage. You know, this is Florida.
Yeah, 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 yeah.
The water has to go somewhere. But literally across the street, there's clearly a lot of owls based on the ruckus they make, and I've seen mom and dad owl raising little owlets
Oh, nice.
sometimes. So it's a wonderful connection to nature as well.
Mm.
Um, but if we think about other levels of biodiversity, um, vertebrates, much as, we love the vertebrates because, well, we are, we are a vertebrate.
Yeah.
Um, the diversity of life outside of that is incredible and really poorly characterized.
Mm.
Mhm.
Yeah, I think of fungi in particular. It's, there's this entire network across the whole North America.
Yeah.
And do they classify it as technically one entity or one organism, or is that being disputed now?
Well, certainly. So a number of fungi are associated with plants. So, you sort of have this network of so-called mycorrhizal fungi that associate with the roots of plants.
Mm.
And some of them can be quite large. I mean, they certainly don't form a network that would be, continent wide, but they're pervasive. Many plants have them, and a fascinating thing, and this is not something I've personally studied, but there are actually parasitic plants that parasitize via mycorrhizal connections. So the mycorrhizae being the fungi that are associated. And some of these can be quite, large in the sense that they're connecting multiple plants. Um, and again, biodiversity of fungi is incredible. And many years ago, I was lucky enough to be a part of the Neurospora genome project back in the day when sequencing genomes was expensive and challenging. um, Neurospora is a wonderful fungus in the history of, well, it's a wonderful fungus overall. It's sort of a nice orange fungus. It's a fire adapted organism. So uh, you the sexual spores are activated by heating, but it also has an incredible history in the field of connecting genetics to biochemistry. Some of the earliest mutants studied where people connected biochemical functions directly to genetics happened in that organism, um, in part because fairly easy to grow in the lab.
Mm.
So, so I will say that, one of my mentors, Don Natvig at the University of New Mexico I was lucky enough to work with him, and he brought me into that wonderful world for a couple of years. So,
Mm.
um,
Yeah.
I still look back on that fondly, even though, my own research has moved away from that.
Yeah, yeah. It's nostalgia's a special thing. I
It's a
just think
powerful thing.
it is very powerful. Yeah, we have such a bias for it, and it just reminds me of when I was young, going in the woods this time of year in autumn and seeing all the fungi. And my favorite part about fungi is their names. I don't know about in the US, but there's in the UK we have such cool names for some of the fungi. It's like the destroying angel, amethyst deceiver. It's just so many cool names. Do you have any cool names for US
Oh,
fungi?
well, I should know more, but certainly there are all these common names that are really that are really cool. But my personal favorite, and I have to admit that I have a somewhat selfish reason for making this my personal favorite, is the hen of the woods,
Yeah.
uh, because they taste really good. So,
Yeah.
you know, um, so certainly, humans have this incredible history in some ways with various fungi in terms of well, obviously many uh, are um, you know are toxic,
Yeah. Yeah.
and some are hallucinogenic.
Yeah. Yeah.
um, and but also many are just have incredible flavor. I wouldn't trust my identification skills.
Neither. Yeah.
I think it's a special person that gets gets confident enough that they
Mm,
go out mushrooming. I'm glad there are people who do that,
mm.
but I was also I'm trying to remember who told me this, and whether it was direct or indirect, but it was a mycologist who was asked something about how people should behave with, their mushroom collections and so forth. And he was like, well, yeah, if you eat them, preserve some of them because, then if something bad happens, we can identify. And to be fair, I had the impression that he was not sort of flippantly saying,
Yeah.
uh, if you eat them and you die, it was more, if you eat them and you get sick, we can help.
Yeah.
Don't eat them all.
Yeah.
You
Yeah.
know? At least that's the spin I'm putting in. Since I can't remember how I even heard this story, I can't attribute it maliciously to anybody.
Yeah. Well, the thing is with fungi as well is some of the most poisonous or toxic ones can look so similar to an edible one,
Yes.
and it's just like so scary. I just do not trust myself.
That's like I said, I wouldn't trust myself either. And then indeed, even with some plants, there are
Mm,
some. Uh, I'm trying to remember some of the things we've seen growing, but there's some yam relatives, and it's sort of like, is this one that's, you know, and the person I was with was telling the ways to identify, and it's like, I just don't know if I trust.
yeah. Yeah. Yeah.
So um, I'm always amazed by how people over human history adapted to different crops and things like cassava that require special preparation, or things where parts of the plant are toxic and other parts aren't. I'm sort of thinking like, you know, how did that happen?
Mm,
Og and Grog hanging out, in the Paleolithic era, going, "Ah, let's try this."
yeah. Yeah. Yeah.
But clearly, humans have, you know, I criticized humans earlier as the asteroid hitting the planets. Humans are also extraordinarily clever, and we figured a lot of things out. Um,
Mm.
uh, you know, uh,
We should be grateful for all the ancestors who were part of
yes.
the trial and error phases of our.
Exactly.
Yeah.
Even in the sciences, I started out by saying science is a team sport. But we also all owe um, all of us doing science owe a debt of gratitude to those who came before us, developed ideas, developed techniques um, that we're using all the time now.
Mhm.
So, it's the same thing on a sort of different time scale. And of course, what we call science sort of adjusts. I tend to think of science, very much in the modern academic framework. Um, where we're hypothesis testing. But, humans to some degree are all scientists. We like to say that kids are always scientists because they want to figure out
Mm,
how stuff works, and they're
yeah.
willing to ask um, the hard questions, the
Mm,
simple but hard questions sometimes. So, um, we're all scientists to some degree.
yeah. And we should cultivate that. We should continue to do that throughout our lives. Too many people lose that curiosity, that spark. It gets drilled out of them as they grow up or when they go through the education system in some areas. And it's a
Yeah,
shame, really, because I think most of us are born with that, if not all of us are born with that. When you're in nursery, you're picking the
yeah.
bugs and looking at the leaves and asking, where did I come from? Just loads of different things like that. I remember asking so many questions as a kid, and I want to continue. That's why I've made this podcast. But it's a shame that we lose that. And I don't know about you, but I guess because you're in academia, it might differ because you have people like that around you. But it's very hard to sometimes have those curious conversations with people because it's not normally where you start off. You don't go up to someone and go, "So, what was the common ancestor of the archosaurs?" That's
Yeah.
not
No,
common small
no.
talk.
And, it is, being in academia is sort of a different world
Yeah,
because, obviously some academic science is, um, is getting closer, is close to translational things that might, down at our med school and so forth, people are doing things that some of which are fairly close to they can actually, help somebody directly. But one thing I always try to stress to people, including students, when I teach my genetics course, which is really my big, um, sort of big course where I have a lot of undergraduates. I teach periodically the genetics. I often teach also computational biology, but those tend to be smaller courses. um, but I try to say, it's really good that we have people doing translational research.
yeah,
That's important. It's helping human lives, whether it's something to deal with biomedical stuff, which is really closer to my field, or other things that directly feed in. But in many ways, the big discoveries are driven by curiosity. Just things that you would never imagine could give a practical outcome. Will suddenly revolutionize a field. Um, which is, I was asked by a reporter some time ago uh, who wanted me to give an opinion on a fossil bird find. I was not involved in the paper, so I was their sort of outside source. And so I gave my quotes and so forth. Um, and at the end of that, she went, "So why does this matter? Why
mm,
was anybody care about a fossil bird from the Cretaceous?" And I went, "Huh. Well, I care because I like birds,
yeah, yeah.
and I want to understand how how they evolve." And then I thought about it for a moment. And I went, "Well, you know, curiosity-driven science tells us how the world works, and understanding that's a virtue.
Mm,
And if we went back and forced every scientist over history who was playing around with some idea to justify what they were doing in terms of a product that might be available in 5 or 10 years, most science wouldn't get done.
mm,
And more importantly than that, for people who are focused on things that are very practical, um, we would miss out on some really revolutionary things that have created entire fields that are now worth, you know, billions.
Yeah.
Biotechnology ultimately grew out of just trying to figure out how life worked at the molecular level. We certainly, the people who were when Watson and Crick solved the structure of DNA um, in the early 50s, they weren't thinking about um, biotech
Mm.
and the amazing things we can now do to manipulate organisms. Or in the UK, Fred Sanger. Um, so you have over there the Sanger Center for Sequencing, and Fred Sanger is one of those rare individuals who has two Nobel Prizes for developing how to sequence proteins and how to sequence DNA,
Wow.
and that changed the world. I
Mm.
mean, my own career would not have been possible without the ability to sequence DNA, and obviously modern technologies. Uh, we can sequence inexpensively huge amounts of DNA, but without that revolutionary foundational work, we wouldn't have that.
Mm.
So um, sometimes this curiosity driven science pays off way bigger than, something that a person who's saying, I see no reason why you should ever fund that stuff because what's it ever going to do for me? Well, it might change the world for your kids.
Yeah.
Might not. I mean, the vast majority of things don't change the world. But here's the thing, we can't predict what's going to change the world and what won't.
Exactly.
It's the fact that humans are curious that changes the world. We go out, learn as much as we can about how the world works. And some of it is just satisfying our curiosity, some of it is something that grows into something revolutionary. So, um, if I can give a little pitch for basic science here, you know, unfortunately, you know, over the years, people have invested in basic science, and I hope that continues. So,
Mm. It's a lot of the most important things, like you said, start with curiosity, and that's also adjacent to play. There's something that the person enjoyed. If they were told by someone above them to do research on this practical thing, I wonder how long they'd actually last before they give up. Whereas if you look at this is not really science, but it kind of is with the Wright brothers and them creating the aeroplane and flying, they failed for years and years and years, but they were curious and it was still play to them and they were passionate about it. And now we take flying commercially for granted. I
yep,
could fly like 8 or 10 hours and meet you in person if I wanted to and could, pay for the plane ticket because of that discovery. And if a lot of other people were trying to create the first aeroplane and failed because they didn't have that passion compared to the Wright brothers and that curiosity and that playful spirit. So if we lose that, I think we probably end up losing innovation and science, right?
oh, very, very much. And obviously, you know, uh, it wasn't long after the Wright brothers that aviation started to become practical. I mean, we certainly weren't
Yeah.
doing transatlantic flights for a while, although, Spirit of St. Louis. But, still, um, that that sort of curiosity is important. And I think one of the things I love about being an academic uh, is um, get to sit in my office and think about problems that I enjoy thinking about.
Mm.
Um, you know, um, and I'll go home and uh, I'm off the clock, quote unquote. But if I'm in the middle of thinking about something that's I'm passionate about thinking about. It's not like I'm gonna wait till you know, 9 o'clock in the morning
Yeah.
when I unlock the door of my office. I'm I'm going to pull out my computer and maybe play with it, see if, or I've even sometimes been known to, an idea will hit me as I'm like going to bed. I open my iPad and write a quick email to myself about, okay, tomorrow morning, this might actually be the solution to what you've been working on.
Yeah, yeah, yeah.
Usually it's about, one sentence fragment, just enough to remind me
Yeah.
of what hit me. And unfortunately, I will admit that most of those sentence fragments, I wake up the next day and go, that wasn't really the solution.
Yeah.
But occasionally.
Yeah. Yeah, yeah, yeah. That's all that matters if it happens once in a while.
Yeah, so that's actually a nice aspect of of working in academia and actually, you know, um, living in a college town. You know, I go have friends over for cocktails or something, and you'll get there are people from various either associated with the university in some ways or something, or you know. Uh, just very curious. And the cocktail conversation turns to things that are sometimes very obscure. Occasionally it has turned to things like you know the genomics of birds or something. So
You're always hoping it does in the back of your mind. You're like,
yeah, I'm sort of
please,
sitting
please.
there going,
Yeah.
I sure hope I get a question. So this is one of the remarkable things about being an academic. And, certainly, uh, I'm glad there are also people who do science degrees that, go on to various industry positions where they are closer to, you know, doing things that, have a direct impact. And I've had students go to other academic settings. I've had students, for example, go to, uh, uh, be a bioinformatician in industry. Um, Different people like different things. And uh, I think we need all of these people. We need people driving the basic research. And, you know, we do need people sitting there doing the brass tacks practical solutions. And that's both practical solutions to things like um, they have a direct impact on, say, human health, or, obviously, as a biologist, I'm far further away from more, tech things, but um, also practical things on the other side of biology, like preserving the environment, you know, um, which, shouldn't really be sort of a political thing because all of us, should treasure the environment. And the reality is most of us do, regardless of our politics. I mean, Florida um, uh, is one of the nice aspects of Florida as a state is you go out of the city and there's just some wonderful wild areas here.
Yeah. Go to the
Um,
glades, the
yeah.
Everglades. Yeah, yeah.
Well, I usually stick north.
Ah, okay.
Not that I haven't been to South Florida, but, there's a lot of wonderful stuff in North Florida as well. Um, and of course, I mostly grew up in the state of New Mexico, the land of enchantment. And, if although very different environment from Florida, anybody wants to go for a nice vacation, um, uh New Mexico story, and they wanna come to America. I mean,
Yeah.
obviously.
Yeah, yeah, yeah.
Um, New Mexico is high recommendation. So
Yeah. I mean, the images of from Ansel Adams and paintings of Georgia O'Keeffe and places like Santa Fe, it just looks so enchanting. It looks incredible. The landscape there, the architecture is
Oh,
is
it's wonderful.
it's amazing. And speaking of your where you grew up and stuff like that, what was your childhood like? You said you grew up in New Mexico, and how did you actually become so interested in genomics? Wait, hold that thought. I'm genuinely so grateful that you've stuck around with us to listen to these fascinating insights from the guest so far. If this episode has given you one piece of knowledge which has helped you understand yourself and the world a little bit better than before, the absolute best way to support us is to pay it forward and hit the share button right now. Don't let this knowledge stay hidden. Please send it to one person who will benefit from this episode. Thank you so much. Let's dive back into the rest of the episode.
So that's a great question. So I was born in upstate New York in Rochester, and my family moved out to um, New Mexico when I was relatively young. So I consider myself a New Mexican. um, and growing up, uh, I don't really remember a time when I wasn't interested in some sort of science. And over the time I was growing up, I honestly wasn't sure what kind of science I wanted to do. I flirted with physics, with biology, chemistry, computer science. Um, one benefit of being a computational biologist is I get to be a not very good computer scientist. So
Yeah.
I get to combine some of those, and
Yeah. Yeah.
fortunately, I work with some very talented computer scientists. So I get the actual computer science uh, um, fed to me a little bit, but um, I really wasn't sure what I wanted to do. Um, and I will say that, I came from relatively, um, working class has different connotations in the UK than it does here, but somewhat working class family. I was first in my immediate family to go to university. Um, And, I'll be honest, I sort of happened into it. I just kind of was taking classes and went, huh, this genetic stuff is kind of cool. Um, and decided to go to grad school. and, and that was all that happened. And even, through grad school, you know, I kept some interest in computing. And one semester, uh, the High Performance Computing Center had some money to support a biologist, and they wanted to bring a biologist in, so they supported me for that semester. Um, but they didn't actually have anything specific they wanted me to do, which was cool. Because basically they gave me access to what at the time was a very powerful computer
Mm.
and didn't really care what I was doing. I mean, I lie a little bit because they had me working with CS grad student on a few things, but it was more sort of I was being a consultant essentially. And beyond that, I was just okay. If you want to do something, uh, here's a computer. You know? um, and N Certainly, I was always interested in that, but it really cemented that interest and allowing me to kind of play, um, I played on defined problems. It wasn't like I just sat there and, you know, did goofy stuff.
Yeah.
I came up with specific questions I wanted to ask. Some of my earliest bird work was done in that environment. Um, but it was really just a wonderful experience to have that. Um, and from there, so you asked me about childhood. Um, in many ways, I was like a normal child. Grew up, had friends. I was a bit of a nerd, hence the Star Trek joke earlier.
Yeah.
Um, uh, some of my friends were nerds. Some of them were, way cooler than me. Some of them were fellow nerds, as I said. And, just developed this sort of interest in science. And like I said, it was very diffuse in part because, um, you know, my, uh, my father, when I was growing up, was a vice president at a bank where he administered their security and investigations department. So very, very practical, making sure, you know, credit card fraud didn't happen and things like that. Um, and, the stuff I was interested in, I think was a little, my parents always indulged me, but I think neither of them really were like, why is our kitten so interested in this crazy stuff?
Yeah, yeah.
And, that sort of drove me into, sort of doing basic science. And like I said, it was really just learning some basic genetics that got me kind of like, wow, this is really fascinating. I want to know more.
Mm.
And from there, uh, did my PhD. I brought in some computational biology starting there. Um, I then went to Ohio State, where I had a wonderful mentor, Eric Grothwald, who's now actually at Michigan State, who's interested in gene regulation, but he wanted somebody who had uh, sort of some knowledge of computational biology and so forth. So um, we worked together on some problems, and from there, um, I was hired at Florida, um, and I've been here ever since. So, um, Florida's again a nice, as I said, it's a nice state in terms of natural areas. Gainesville, being a college town, uh, if if you're just curious about stuff, uh, move to a college town.
Yeah, definitely, yeah.
Because you'll meet, even if you're not an academic, you'll meet lots of people um, who are, or who are just curious.
Mm.
Not that there aren't other places too, but
Yeah, it's just the concentration's higher, the ratio
exactly.
is much higher than yeah, I understand. And, going ahead of time a bit now,
Yeah.
you were the co-author of a paper sequencing 48 bird genomes, am I correct?
Yes. Yes.
Yeah. So what was some surprising new insights that you gathered about birds that most people won't know from that paper? And
ah,
how did you actually sequence all of these different bird genomes?
so I'll one thing I'll say is we built on the initial 48. There were over 300 were published in about now 5 years ago. So this is an ongoing effort, and I'll especially give a shout out to Goji Zhang at Zhejiang University. I always apologize to my Chinese colleagues for my inability to say anything sensible in Chinese, as well as Eric Jarvis and Tom Gilbert, who uh, were instrumental to that. Um, in terms of how you sequence, modern sequencing is actually sort of wonderful. It's a fast moving technology. So I'm going to oversimplify a little bit because, techniques, are constantly changing. But really the innovation that happened in, you know, the mid aughts, to late aughts, early 2010s, was the idea that you could sequence many fragments of DNA in parallel. And it greatly reduced the costs. And the cheapest technology is still the least expensive technology is still one that sequences relatively short fragments of DNA. But there are now ones that can sequence long fragments. And basically, what you do is you chop up a genome. And to some degree, when you isolate DNA, it comes a little pre chopped. And some of the things we've done recently have been out of museum specimens, birds that were collected as much as, 100 years ago. That DNA is pre-fragmented. It's been sitting in a cabinet for 100 years.
Wow.
Um, so, um, but you take DNA that's been chopped up and you sequence a bunch of fragments of it and you use a computer to put it all back together.
Wow.
And those methods are not perfect. There's always going to be holes in it, although it's getting better and better. There are now some so-called telomere to telomere genomes coming out, which the telomere is the end of a chromosome. So telomere to telomere is the whole thing if you take it seriously. Um, but the I'm gonna give you um uh uh the the thing that that at least I am most excited about. And I'll give you a little backstory because it's unfortunately a complicated finding. Um, the backstory is um, if we think of any group of organisms, I've talked a lot about populations. Populations are the units of evolution. And of course, populations themselves have variation. And so if you go back to the common ancestor of, say, humans and chimpanzees, there were there was genetic variation there um, in that common ancestor. So when humans and chimpanzees split, 5 or so million years ago, the exact genes that got passed on were a sample out of that early population. There's an important consequence to that, which is although humans are more closely related to chimpanzees than they are to gorillas, if you look at individual genes, it's only about 70% of our genome. I'm rounding, but about 70% of our genome are the genes are chimps and humans more closely related to each other. If you look at the other 30%, about half of those, if you were to trace the history of those genes, humans and gorillas are more closely related for that specific gene.
Mm.
And for about 15%, chimps and gorillas are more closely related to each other for those specific genes. So it's sort of a 70-15-15, give or take. Um, so this is a phenomenon called incomplete lineage sorting. The idea is the common ancestor of humans, chimps, and gorillas had variation. Gorillas split off. Then variation remained in the evolving population. And then we had a split between humans and chimps. And how exactly that variation was spread out was itself somewhat random. And there's a
Mm.
lot of complexities here, but to a good first approximation, if we look at most of the genome, it's just relatively random. It's all driven by genetic drift, this phenomenon writ large. And I won't go into a lot of there's an entire fascinating area here. In fact, I gave a academic talk that I entitled something like "Incomplete Lineage Sorting," an underappreciated source of conflict among genetic histories, which if you were in the field in academia, you'd be going, oh, everybody talks about incomplete lineage sorting. And despite that, I think it's still, there's riches to be mined out of that area. So let's take the human chimp gorilla story and apply it to birds. So when the non-avian dinosaurs went extinct and a small number of bird lineages made it through that mass extinction, one group of birds really underwent an incredibly rapid period of speciation. This is so-called neoaves. And basically, if you see a bird and it's not one of the barnyard birds, duck, goose, chicken, turkey kind of thing, or one of the paleognaths, so the ostrich, emu, cassowary, if it's not one of those, it's a neoaves. So
Passerines, right?
basically, if you go outside, whether it's a little songbird, an albatross, a penguin, they're all neoaves.
Is that the same as passerines, or is that slightly
Uh,
different?
so the passerines are the passerines are so-called perching birds, and the majority of passerines um, are the songbirds. The Aucine passerines is the songbirds. There's also a sub-Aucine lineage, which is pretty diverse. A lot of them in South America, some elsewhere. There's some nice old world sub-Aucines as well. There are more than half of birds, but the neoaves is passerines plus parrots plus falcons, hawks,
Mm.
um, you name it. If it ain't a barnyard bird or a paleognath, um, it's a neoaves. Now, the major lineages of neoaves split very rapidly. And it's the time between speciation events that determines the amount of incomplete lineage sorting. So the reason why only about 70% of human genes have the human chimp gorilla history is because of the amount of time between the speciation of gorilla versus human chimp and the speciation of human chimp. So if you made that distance between the two of them smaller and smaller in time, you would have more and more incomplete lineage sorting. But it's nice because it actually makes a prediction about, if you look at the genome, how much of this do um, how conflicting are the histories. If you pop them into a computer and try your best to reconstruct the histories, how much conflict do you get? And the answer is, of course, a lot of conflict. And um, uh, this is work that I really need to give a shout out to my colleague Saivesh Mirarab who's an incredible computer scientist at UC San Diego. Um, what we noticed, along with some other colleagues, too many unfortunately to name, um, uh, was that there was a particular segment of chromosome 4. We're mapping everything to the chicken chromosome. So we're numbering our chromosomes the same way we number them in chickens. There's been some chromosomal rearrangements, but we have to reference somehow. We found a segment of chromosome 4 where there was almost no conflict. So the incomplete lineage sorting was happening everywhere in the genome except that one place where every gene pretty much agreed with every other gene in that one region,
Mm.
which sounds really weird.
Yeah.
We predict this incomplete lineage sorting phenomenon Why does 98% of the genome have it? 2% in one particular part of the genome doesn't. And the short answer is part of what drives this incomplete lineage sorting. I talked about the fact that it's related to the variation in populations and how that is randomly passed on. Well, of course, individual genes are sort of mixed up every generation. So, we all carry, humans were diploids. That means we have two copies of each chromosome. Let's leave the sex chromosomes out for now.
Yeah.
But, of the non-sex chromosomes, we have two copies. We get one from mom, one from dad. And same is true for birds. They're all diploids. They have different sex chromosomes, but we're leaving the sex chromosomes out. Um, every generation, the gametes are produced by reducing the genetic content from two copies of each chromosome to one. And then when two gametes come together, when a sperm and an egg come together, sperm is carrying one copy of each chromosome, the egg is carrying one copy, and we get a new diploid. Okay. However, every generation, when producing those gametes, the chromosomes from mom and dad in the individual making the gametes are mixed up. This is a process called recombination. Chromosomes line up, they cross over, and segments swap. Since that's happening every generation, each gene is being inherited somewhat independently. If they're super close to each other, only a few base pairs, um, then they're pretty much always inherited together. But even within a gene region, and I'll just to show you the scale of this, most genes in vertebrates are split up into the part that encodes a protein called exons and intervening regions, which are non-coding. And they're removed during gene expression, the intervening regions. These are called introns. So my wife, Rebecca Kimball, and I have been studying a gene region where there are some specific biochemically interesting changes. And I won't go into all the biochemical details because that could be a whole academic talk. But the changes occur in two different exons. So they're separated by a non-coding sequence and then put back together to make the final protein. Here's the cool thing. We see evidence that the evolutionary history of those two regions is actually different. So 30 million years ago, the mutations happened independently. And during a mating, they were put together by recombination. And
Uh,
they're within, 1,000 base pairs, base pairs being the ACGT that
yeah.
makes up our genome. Um, so even at that small scale, everything's mixed up. So what did we discover on chromosome 4? Well, it looks to us like what happened is about the time when the non-avian dinosaurs went extinct, there was a rearrangement in on chromosome 4, such that part of the chromosome flipped around. Now, when you have something like this, it's called an inversion because you flip part of the chromosome. Most of the chromosome lines up and can exchange genetic information by recombination. But in the flipped region, it doesn't exchange. And now we as humans, in human populations, there are small inverted regions that differ among individual humans that are in the population. So this is not something unique to these birds. Um, Pretty much look around at many organisms. If you sequenced very deeply, you would probably find a lot of inversions. This particular inversion on chromosome 4 was big. It's over 20 million base pairs, and it probably persisted for several million years in both forms. So some birds in the population had, um, both forms were, we often refer to this as segregating polymorphisms, which means polymorphisms, multiple forms, and genetic segregation is when the gametes are being formed. So what that would really mean is if we could go back in time and sequence some of these birds, we would find some individuals that had both forms, the original form and the flipped form. Some birds would have both flipped, and some would have both original. During that time, the flipped and original forms were not recombining. And then by genetic drift, one or the other form was fixed in certain lineages. So the what we believe to be the flipped form went on to become fixed in the common ancestor of grebes and flamingos. So everybody knows what a flamingo is. And if you don't know what a grebe is, if your listeners don't know what a grebe is, Google grebe and grebes dancing because you'll get some beautiful videos.
Yeah, I've recorded it
uh,
before. Yeah, it's amazing.
and um, and the other group that has the flipped form includes doves, and a few other lineages that I won't name because they're not commonly known. Uh, Birders will probably know them, but um, so for that region of the genome, that 20 million base pairs um, of chromosome 4, pretty much everything in it, if you were to reconstruct its evolutionary history, puts doves and their close relatives sister to grebes and flamingos. If you look at the rest of the genome, it's a mixture. And it turns out that if our best estimate of the species tree based on whole genomes um, is that the first lineage within neoaves to split off was the ancestor of grebes and flamingos, and then everybody else. There aren't that many grebes and flamingos, so it was between a lineage that really didn't speciate much and 95% of birds. So, um, I think that's an absolutely fascinating story because it connects a process that happens every generation, this recombination and mixing of genes with um, something that is still visible 65 million years later or so. And in my mind, we learned a ton, and I could probably spend an entire day regaling you with stories of things we learned from bird genomes. But at least in terms of the thing that's nearest and dearest to my heart, the fact that we can preserve a signal of the recombinational history of the genome for more than 60 million years, that is super cool. So,
Yeah, that's
and I hope I convince you it's cool.
it's yeah, the fact that you can go that far back in time,
Yep.
and it's just mind blowing. It really is.
And it was also a lot of luck there. You
Mm,
know, one bit of luck was the way we indexed genes. So the uh, effort that produced a genome scale tree of more than 300 birds was led by Josephine Stiller, and a number of other people who I've already named were involved. But um, she had indexed all the genes based on where they mapped in the chicken. So they were just given numbers. And she was trying to pick them about every 10,000 base pairs. And then when we were looking at them, we saw this weird signal uniting, strongly uniting doves and their relatives with flamingos and grebes. And then we looked at the indices that, the numbers assigned to the genes, and it was like, wow, they're contiguous. Why?
Yeah.
And that's what started this analysis. So I think being attentive to details is, I guess, if there I don't know how many young people, in high school or something are listening, but or even younger, don't lose your curiosity. And if you want to be a scientist, look for little things because sometimes they turn out to be super cool.
Mm.
So,
Yeah. Yeah. Speaking of science and higher things, as a few final questions. First one is, do you believe in God? Are you religious?
I'll just say no. As atheism is fairly common in biology. Now, uh, I really appreciate a lot of people. Uh, one, I think religious freedom is important. Um, and, I always joke about loving the fact that the world is filled with uh, Bono Christians. Although I've gone to U2 concerts, I've never personally met the band,
Yeah.
but um, so I don't know what any of them are actually like at a personal level, but I sort of admire the fact that um, Bono has said that God will judge humans harshly if we don't take care of each other.
Mm.
And uh, my big hope is that rather than dividing us, history, religion, and people of faith take that attitude.
Mm.
Uh, you know, obviously, as being an atheist myself, uh, I've periodically considered going to a Unitarian church because uh, as one person told me, that's church for people who don't believe in God.
What is it like? What's the Unitarian
well,
church like?
well, I've met some Unitarians and talked to them, and I think many of them do believe in God. So it was a joke somebody told me, obviously, but I think it's much more ecumenical. Now, if you believe in a particular creed, I'm certainly not going to disrespect anybody as long as their faith drives them to want what's better for their fellow humans
Mm.
and the rest of the world. And, of course, one of the things that does unfortunately divide us is we all have our own ideas about what's best for our fellow people.
Yeah.
And I do like to believe, despite some of the divisions among religious people, that the majority of people do want what's best for their fellow people. And when really faced with hard decisions, will hopefully adopt a more ecumenical attitude.
Mm.
Whether that's true, um, I guess I do have faith, which is there's a lot that we could look out in the newspapers and see and say that what I just told you is absolutely not the case.
Yeah.
But I have faith that um, when push comes to shove, it will be. So,
Mm. And do you think you can believe in evolution and God
oh,
simultaneously?
absolutely. You know, Science tells us about how the natural world works. It doesn't tell us about things that are beyond the natural world. And indeed, there are one of the probably most famous evolutionary biologist, Theodosius Dobzhansky, was a man of faith. Um, and there are many evolutionary biologists who, um, obviously some are atheists, some are agnostic. And indeed, I find the distinction between atheist and agnostic a little fuzzy because you can always, if you don't have a little bit of question in your mind, you're not questioning enough.
Yeah,
Um,
yeah,
but there are also people of faith. Obviously, there are some faith propositions that are incompatible with evolution. And I would argue that that faiths that are dependent on a particular genomic history, for example, which of course Noah's Ark would drive.
yeah.
If you really went down to the animals going two by two onto the ark,
It's a bit too
we would
recessive.
see that in the genomes.
Yeah.
um, and we don't. But if we look to these stories of faith and so forth, um, it's possible to believe in the Big Bang, in evolution, et cetera, et cetera. And that's where the science points. Um, and it's possible to also believe in God or have other forms of faith in the rest of humanity. And
Mm,
without that faith, the earth, the life is probably a little hollow.
mm,
Whatever that particular faith is. I think a
mm.
person who drifts into nihilism and doesn't
Yes,
believe
that's
in
what I was
their
gonna say.
fellow human, that's dangerous.
Yeah, I was going to mention that, the nihilism, when you don't have some sort of faith, and that,
Yeah.
gets me to ask, what is the meaning of your life? What is your faith? What is your compass?
And that's a good question. And I think it's just to sort of be with people you love, be with people you like. Try to make the world a better place however you can, whether it's a small way or a bigger. Some people are able to do it in a big way. Some people are only able to do it in a small way. It's, I think, um, uh, just trying to remain curious about how the world works, trying to be accepting of other people's beliefs. Um, you know, uh, I think, unfortunately, and I'll just throw out one small thing in the last minute. I worry that uh, social media has done us poorly by allowing all of us to share our views without teaching us all um, to forgive each other when we express a view that might be controversial, might make you angry. Um, I, normally, when I was a kid, you joke around. Sometimes you say things that are maybe a little off. Not everybody hears it. Only your friends hear it, and they
Yeah,
know who you are.
yeah, exactly.
And uh, but now people can spout off about everything, and we see that in saying horrible things about person being assassinated in America. Um, you know, Regardless of whether you agree with somebody or you don't agree with somebody at a human level,
Mm,
you have to say, I don't delight in anybody's
yeah.
being harmed and certainly being killed. So,
Mm,
um, I think that's something we're going to have to overcome is um, the fact that we all say stuff. And now, instead of saying it only to our friends who we can kind of gauge how they're feeling and then, maybe smile and everybody acknowledges that they know everybody and where they're
Yeah.
coming from. Uh, we're saying it to millions of people.
Yeah.
And I think maybe what we need is some forgiveness. If somebody
Mm,
says something a little off, maybe you say, I just need to let that go,
mm,
and just go back to trying to be the best you can be. So
mm.
I guess that's
Yeah.
my view.
Yeah. 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 And it's interesting what you say about saying things that you would only say to your friends. I used to play a lot of Xbox, and we'd have the private chat parties. And
Yeah.
there's this meme that, like, if there's a clip of people, like a bunch of like young males being handcuffed and walking to prison. It's like, what would happen if the Xbox chat got leaked? And it's like we all have
Yeah.
these thoughts and these discussions, and it is play until you say it to someone you don't know and millions of people you don't know without thinking and being impulsive about it. And it's not like before social media, these thoughts weren't uh, happening. They were, but it just wasn't leveraged to millions of people.
And you also weren't saying it to people, as long as you have a group and everybody knows everybody
Mm.
to some degree, you know, would you want the meanest joke you ever told in your life to go out to a million people?
No.
No, and most of us wouldn't say those. And the worst part, in my opinion, and I'll just sort of close out a little bit with this, is people say things on social media that they would never say to somebody's face,
Mm.
you know? And that's another thing we should potentially do, which is before you type something, ask yourself, would I walk up to somebody and say this?
Yeah. Yeah.
Um, and, there probably are people who are not, a little, they're angry or whatever, and they would say nasty things to people's faces. But most of us aren't that way.
Yeah.
And maybe we need to start to think differently and both think twice before we say something that's a little nasty and or put somebody down for things they sincerely believe, and also forgive people who trespass against us in that
Mm.
way. So
Mm.
um, that's my unscientific view.
Yeah, yes, yeah, no, there was some religious connotations in that. Don't trespass. I liked it. Um, yeah, and final question: if you could give your 20-year-old self one piece of advice, what would it be and why?
Oh, that's a hard one. I think the biggest one would be don't take life quite so seriously. You know, it'll all work out. You'll find ways to be happy. You'll go on a journey that, um, takes you places you didn't expect, and somehow it'll all work.
Hmm.
And I think that's probably the advice I'd give myself because, I was pretty tightly wound when I was 20. Because,
Mm.
then I was like, oh, I've got to accomplish all this stuff.
Yeah, yeah,
And I didn't even know really what I wanted to accomplish. I was just wound up.
yeah, yeah, yeah. I mean, as a 21-year-old, I have those drives as well. So that speaks to me. So thank you.
Okay.
Edward Brown, ladies and gentlemen. Edward, really, really appreciated this. And yeah, I learned a lot of different things. And I'm glad we went through a lot of different topics. And I understand that a lot of it wasn't your direct field, but you still had some fascinating insights to share with us about those adjacent topics. So I really appreciate it. What are you currently working on?
Um, so we're currently trying to build well, we're working on a number of things, but the biggest one is we're trying to produce a tree, uh, evolutionary tree of all birds, using. I mean, people have done this, synthesizing data. We're trying to build up a data set that will give us all 10,000 birds,
Wow.
and a lot of and a lot of data sampled from all of them. Eventually, we'll have genomes from all of them, I'm confident.
Yeah.
But um, hopefully, you'll see that out soon.
Awesome. And where can people go if they want to find your work that's already out there?
Actually probably just Googling me, especially my Google Scholar page.
Yeah.
If you're interested, you'll see some of my work. I really need to update my UF web page as well. I let that get uh, out of out of date.
Yeah.
But just do a little web search. You'll see some papers. Um, Most of them are pretty technical, but hopefully some of them are, I think they're all interesting. And some of them I think can be appreciated even for the non-technical.
Awesome. Yeah. I'll leave a link to the Google Scholar page in the show notes for everyone to check out now. So everyone go check that out right now. Thank you so much, Edward. Really appreciate you.
Okay. And thank you for giving me the opportunity to discuss this with you. It was great meeting you.
Yeah. Awesome. If you've made it this far, you're likely not just a listener, you're ready to be the host. You felt that sense of intimacy and connection with our guest today, and you would like to get the same on your own show and start your own dream show. But you're worried about landing dream guests and all the complications and the mess that is often spoken about when starting your own podcast from scratch. You think starting a podcast requires a huge budget and a whole team, right? That is absolute nonsense. My pro remote podcast blueprint is the ultimate key to skipping the outdated advice and building a high quality, high impact show, even for under $100. It provides the proven strategies and AI templates that I use to this day that you will be able to use to reclaim your time and creative control. And this is a strictly limited opportunity. Don't dilly dally because there's limited spaces. We've already got one student, Rob, who's joined. That means there's only a couple more spaces to get the Pro Remote Podcast Blueprint course half price. And you need to act very, very fast because this is a limited offer and you're very, very early. So if you want to get the early bird discount like Rob has and get the results by watching the course that goes through the exact step-by-step blueprint that I use to this day to create these episodes that you've just watched and get to the success and become Europe's fastest growing under 21 podcaster, and also join live weekly calls with me and use the AI templates and email templates that I use every single day to get the results and just get all my secrets without getting any of the mistakes and failures that I've made, then top link in the description is for you. And if you're ready to launch your own show and get personalized one-to-one help from me and copy my blueprint, which no other podcaster has shared, then I'll see you there. I'm rooting for you. Goodbye. Hope you enjoyed the episode too.